cnidarian part. So this is my first love. Actually, want to guess what it is? Anyone know what it is?
It's a siphonophore.
Indeed, exactly. That's a siphonophore. So this is actually show the basic plan of a siphonophore. They have a very, very tiny float, which is in the head of this animal. You probably don't see that. And in the middle part of it is their swimming body. They use jet propulsion and squeeze water out to swim. And then the long tail of the siphonophore is their predation organ. All the needle cells are there. And it's also their reproduction part too. So just like the definition of species become vague when you are talking about bacteria, the definition of individual also become very vague when you are talking about siphonophore, because the whole individual, the whole siphonophore you see here is actually many individuals living together, but they kind of do different function, a little bit like our organ or our cell, but each of them are independent individual. So we'll talk more about that. So that's what I just talked about. It's called polymorphism, which means each individual have different outer appearance and function differently, but they all live together. And there are three major parts. One is the pneumatophore, which is the head here, the head of siphonophore, and the function of it is to flow, to maintain the buoyancy. And the way they do that is they have nitrogen organ to maintain the buoyancy. And the middle part of the body we call nectosome, and the function of it is to swim. And the way they do that is just to squeeze water out. So it's just like Jacqueline just mentioned, they do jet propulsion. And the tail of the siphonophore, called xyphosome, is actually a cluster of reproduction and feeding organ. And all the individuals are connected together through stolon, a little bit like our blood vessel. So there are three major parts of siphonophore. First part is physonecta, which is the traditional siphonophore. You have float, the swimming bell, and then the reproduction and feeding part. But some siphonophore have a very developed float, and they use that to swim, and they no longer need the swimming bell. So the middle part of the swimming bell, the nectosome, disappear. And there are some siphonophore do the opposite way. They no longer have float. Instead, they have an oil drop inside of their body to maintain their buoyancy. And they have a big nectosome, so they could swim much faster than the others. And they have a traditional asexual and sexual alternations reproduction cycle like the other jellyfish. And their life cycle are all very similar. The fertilized egg will develop into protozoid, and the protozoid will further develop into zoid, which will asexually budding to a whole colony. And then the tail of the colony will shade when it grow to long certain stance. And then the shaded tail will turn into eudoxid, which is a sexual reproduction individual. And that will release egg and sperm and then do the cycle again. Siphonophore basically do that. And their fishing, their feeding mechanism, just like Jacqueline mentioned, is ambush predator. They literally fish their prey. Like the siphonophore here, they have a very, very, very long tail, and each tail have a very, very, very long tentacle. And some of them have a bioluminescent, and many of them have bioluminescent actually, and they will twist their tail and try to mimic copepod to lure their prey. Like here, let me show you, this is really cool. So this is a siphonophore twist its tail, tentacle. It just look like a copepod jumping. Yeah, so when it's a very amazing ambush predator. Here is the siphonophore review. I encourage you to watch that after the class, but we are not going to see that. And since right now it's about 1 hour and we finish the cnidarian part. So I think we'll take 5 minutes break from now, and then we keep going. Now we are going to talk about ctenophore, which is the subject I'm studying right now. For students who took zoology or biology, evolution, something like that before, when I learned it in undergraduate, my teacher told us that sponge is the baseline of animal. Guess what? Not anymore. The new study, really new, this year, 2014, from Nature and Science, a lot of hot research about it, and they find out that actually ctenophore is the earliest sister lineage of animal group. And then it's a totally different system from all the other animals. So I think that's a very strong evidence to prove that actually ctenophore are aliens. They're just so different. I'm going to talk about how come they are so different. And people used to think they are one kind of jellyfish. They used to belong to cnidarian, not anymore. But I'm going to talk about why they are not cnidarian anymore. Well, the basic reason is because they don't have stem cell, they don't have needle cyst. Instead, they have sticky cell called chloroplast. So they don't sting, they're sticky. And then, and then all of the ctenophore have combs, like tenies. It's an array of cilia. And they use that cilia to swim. And they also use that cilia to create a feeding current to move particle through his mouth. And it basically could go through two big classes. One is the class with tentacles. One is the class without tentacles. And in Chesapeake Bay, we have one ctenophore from each class, which is ctenophore miniopsis. We see that a lot during the summertime. Another kind is called ctenophore burrowii. We see them usually around this time, fall and winter. And they have no tentacles. We are going to talk more about it. So like I say, ctenophore, they don't have stem cell, they have sticky cell, and they have a row of tenins. And all of them are holoplankton, so they don't have benthic stage. They don't need to settle on some substrate to grow polyps. Their whole life cycle are planktonic. And they are hermaphrodite, so they don't need to find another individual to reproduce. But they have similar things with jellyfish. They also have stateless, which is like their balance stone. And the way they do that is there's a tiny stone and in a bowl full of cilia. And when they swim in different directions, that little stone will roll over the place and then press different part of the cilia of that bowl. So the ctenophore could know very well that which angle, which direction they are doing. And many of them have bioluminescence, which is the hot study topic in ctenophore too. And like I said, their life cycle is comparatively simple. They don't have benthic stage, and it's kind of direct development. And very amazingly, is their high fecundity. Each of them, if food and temperature is good, could reproduce 2,000 to 3,000 offspring every day. That's why they could bloom in such short time. Just you only need one individual and good food and good environment. That's enough. And because we have miniopsis ctenophore in the Chesapeake Bay, so I will talk a little bit more about their life cycle. So actually, ctenophore have two long tentacles when it's still a baby, but that tentacle will gradually disappear when it grows, and instead it will have a two lobe. It actually gives it advantage on feeding, because with its cilia, it will create a feeding current, but the feeding current only bring in comparatively small prey, like microzooplankton, dinoflagella, and cilia. But since they have lobes, they could function as a pair of hands, so they could grab food, bigger food, like copepods. So they actually eat both ways. They could eat copepods and they could eat cilia and dinoflagella. That gives them a very good flexibility when talking about feeding. And their major predators are sea nettle and burrowii. Some fish also eat a lot of tinafol like butterfish and sunfish, and unfortunately we don't have sunfish in the Chesapeake Bay, and we don't have too many butterfish either. So the major prey of tinafol in the Chesapeake Bay or Miniopsis are sea nettle chrysiola and another tinafol burrowii. And this is how burrowii feed. Burrowii, it has no tentacle. It literally is a swimming mouse. It's a very flat tinafol, and it could swim very fast. And when it swims, it actually closes its mouth. And when it sees its prey, it opens its mouth and swallows the whole thing and close its mouth. I could show you how it preys. It's really, really cool. So this is burrowii. Like you see, it's flat. And then when you eat another Miniopsis, so here is the Miniopsis. And then encounter still Miniopsis. Let's see if I could fast forward a little bit more. Here. Okay, here comes the burrowii. Close mouth, open mouth, swallow the whole thing, close the mouth. Yummy. Yeah, we could see that. I think there's still more coming out. We could see that again. Yeah, swallow the whole thing. Yeah. So yeah, burrowii is a very vicious predator of Miniopsis. In any ecosystem that you have burrowii, you probably don't see too much tinafol brood. They are really good at eating all the tinafol out. But that's a problem, because in the Chesapeake Bay, the two major predators of tinafol doesn't really control the tinafol population anymore. First, we have chrysiola decreasing, and burrowii doesn't bloom at the same season when tinafol bloom. So as the figure here, you see, we used to have many sea nettles, and then they decrease. And then at the same time, the Miniopsis population increased. And Miniopsis is a very efficient predator. Like I said, they have feeding current and they have lobes, so they could prey on both small prey and big prey. So their feeding efficient is actually stronger than chrysiola. So their cleanse rate on copepods is higher, and when Miniopsis increase, copepod population decrease. This is actually a very good example of trophic cascade. Trophic cascade means that the relationship between prey and predators beyond just these two prey and predator. So in this case, it's not just about sea nettle prey upon tinafol. The influence actually go all the way to the food chain. It also influence copepod, and copepod prey on phytoplankton, so that influence the population of phytoplankton too. So as you see here, when we have more chrysiola, it suppresses the population of tinafol, and it actually kind of release copepod from the predation pressure. So like right now, recently we have more Miniopsis than usual, and because they are released from the predation pressure of sea nettle, and as a result, the population of copepod decrease. Why is that? There are three major hypotheses. One is warming, because tinafol doesn't have a benthic stage, so it responds to warming so much faster. They don't need to budding into polyp and stropulation and turn into ephora and wait until they go to jellyfish. They just reproduce and then grow into adult. So they respond to warming situation faster. And also they tolerate lower salinity. So when you have a warming winter, you usually have more freshwater input to the Chesapeake Bay. And also another major reason is eutrophication. Eutrophication change the zooplankton structure. That make the smaller zooplankton dominant in the ecosystem. And like I told you earlier, tinafol, they have a feeding current that is very good at feeding small prey, but sea nettle doesn't. So, and also eutrophication lead to the bottom hypoxia. And tinafol is better tolerant hypoxia than sea nettle, so that make them survive. Another another hypothesis is oyster decline. Wanna guess why oyster decline will influence sea nettle decline?
Competition for prey.
Not the major reason. Think about their basic life cycle difference. What part of the life cycle tinafo doesn't have, but sea nettle have to finish the life cycle? I will just tell you the answer. That's the basic stage. The sea nettle have to have the basic stage, and their polyp like to settle on oyster shell. So when oyster is massively removed from the ecosystem, that impact the sea nettle population. But for tinafo, doesn't matter. They don't have basic stage anyway. So here is the figure of Noah's. The blue line is oyster, green line is sea nettle, and you could see that they decrease at the same time. And it's another reason, like Jacqueline just, another example Jacqueline just mentioned earlier, like in Taiwan, there's a lagoon, and we used to have oyster raft. And when we have oyster raft, there are also moon jellyfish bloom every year, constantly, regularly. And then one year, government decided to put that lagoon into a protect zone. So they remove all the oyster agriculture. And the next year, moon jellyfish just disappear. They actually go back and measure the zooplankton in that water. Actually, zooplankton increase, so there is not enough food, and the temperature and salinity doesn't really change at all. So I think it's very possible that oyster decreasing have something to do with sea nettle decreasing.